Hello, Beth.
Hi, how are you?
Peachy Keen. Good to see you.
Always lovely to see you. Always a lovely day when it's Ask Erin Dear Beth Day.
Love it. I agree completely. Yeah. So I wanted to talk about cell painting again. Because,
I mean, it's fabulous. Why not?
it's fabulous and fun. And we talk about it a lot, and we'll drop, check out show notes. We can link you to other episodes in which we have talked specifically about cell painting. But this time, I wanted to talk about a new sort of meta resource that I put together that I thought people would find helpful when wanting to dive in and become more expert at cell painting.
That seems super useful.
Yeah, so here, we have the Guide to Cell Painting for Biologists.
It's so
Uh,
colorful.
Oh, it is. I was, pretty excited when I figured out how to add in that rainbow gradient. It's here at simonylab.github.io/guide to cell painting. And, we'll have the link, show notes, and all that. But basically put this together sort of for biologists, hence the title. Um, so people coming from a, more wet lab biology, less strictly, computational math expert side, coming from the biology side of things, wanting to know more about cell painting and how to learn more about the different components, maybe how to run it themselves or how to dive into some individual part. So, um, each question we've got here, it will say sort of I ordered it in kind of increasing complexity. So if you find a question here and, work your way that's not chronological. Work your way
Down the
numerically.
page.
Down the page, that's a good way for it. Work your way down the page because they get kind of increasingly in the weeds as you go down. So maybe if you're wanting a giant overview, just look at the first resource mentioned at each of these. And hey, actually, quite often the first resource is like, go check out an Ask Erin Dear Beth episode. Because for those regular watchers of our podcast, you know that these are kind of more, one of the more casual resources that we
Mhm.
produce, but hopefully kind of an easy way to get an overview of things. And then as you dive in, you then work your way into, again, more and more technical detail. So it's arranged kind of by question that you might be asking as a biologist, even starting with like, hey, what is cell painting? I keep hearing this thing. Tell me more about it. I would like to know what this thing even is. Then moving into what can cell painting do? So here, some resources about the applications for cell biology that are common, and what kind of questions, biological questions, can be asked with this kind of data, coming, and some examples of couple reviews, talking about linking you to a variety of cell painting projects, and the sort of diversity of biological questions that they're able to answer best. Um, for those people who are wanting to perform the cell painting assay in a wet lab on their own, we like the very first question we are often asked is, "Hey, can I change this at all? Like, how is that okay?" The answer is a giant yes, and we haven't asked Erin Dear Beth on that.
We sure do.
Um, so, start there, but also a lot of details about, yes, how you can do that, and different examples of how other people have modified this assay to meet their own particular needs, whether that is their own microscope setup versus their own sort of more tailored bit of biology that they're wanting to dive into. Um, how to do image analysis on a batch of cell painting. So this is definitely our, biggest chunk of resources here. But this is, what we do, what we are the world experts in, to toot our own horn a little bit here. What we do most often in our lab with cell painting data is really focused on the image analysis. And we have done image analysis on terabytes and terabytes and terabytes of
We're probably
cell.
closing in on a petabyte of cell painting data.
Yeah, yeah. So we've touched a lot of cell painting images, and have some pretty well tested ideas about how to dive into doing image analysis in cell painting. So here's a bunch of resources that can help one do the same thing. Um, okay, so image analysis is done, and there's image features. How do you turn those into profiles? So part of the cell painting assay is taking, finding individual cells and taking hundreds to thousands of measurements on each cell. How do you get from individual measurements of each cell into a single mathematical description, a profile is what we call it. You can call it a vector, like different ways to describe it, but
Mhm.
how do I take all of these single cell measurements and turn them into a description of each perturbation that I have in my sample set, in my data? Um, then once you have those, how do you find biology in them? What do you do? Because we're not doing all of this just for the love of crunching data. Like we want to ask biological questions.
You don't just love making spreadsheets for the sake of having spreadsheets?
Well, I mean, okay, so sometimes I kind of do, but I understand that that's not most people's happy place. Um, Also, I do want to point out that one does not need to do all of this on their own, right? So we've talked about the cell painting gallery a bunch, through various episodes of Ask Erin Dear Beth, and it is a public repository that we maintain with, again, hundreds of terabytes of cell painting data. So you don't have to do your own image analysis. If you have a biological question, you could just jump into this point of, hey, I'm going to pull profiles from a data set that somebody else has made, but I want to start exploring the biology because for sure there is unexplored biology in all of these published data sets. While the researchers that initially made them and dove into them may have found something interesting, there was guaranteed to be a whole lot more in each of these data sets that is unexplored and also exciting and valuable to other researchers. So y here's how we just start exploring biology in these profiles. And if you have a specific area of biology you're particularly interested in, there's a lot further that you can go deep diving than this. But this is just a starting point to help you kind of look for interesting tidbits that you might want to dive into. Um, Cell painting features. So image analysis that we do with Cell Profiler outputs, again, I said hundreds to thousands of features, and each of those has a name. And this set of instructions here help you understand what was actually, this number I have uh, in this spreadsheet, in this row and column, like what does that number mean? What was it actually measuring based on the column name? And this will help you understand that. Where do we find published cell painting data? If I don't want to do all of the the lead up wet lab work, I'm just a biologist that's coming in and saying, "Hey, I have a question, and I want to see if these resources other people have created can help me answer my question." This is where to start looking. And finally, um, what else can I do? Help. I'm still confused.
Mhm.
And have in here the forum. We also mention the forum a lot. We even have an Ask Erin Dear Beth episode about the best way to write a question on the forum. If
Mhm.
you do want to join our community and ask for help. And, a link to ways that you can contact us directly
Mhm.
to see if we can be helpful to you. So
I
that's the
think there's
gag.
one, I don't know if it needs to be on this thing or not, but you can also contact us for the Ask Erin Dear Beth question submission form and submit questions there.
Absolutely. I will go add that to our to this resource as soon as we're done recording.
Mhm.
Brilliant idea, Beth. Yeah, you can absolutely just ask us and we'll answer it on the podcast. And
Mhm.
oh my gosh, that would be fun. So yeah, that is the overview of this resource. This resource of
Mhm.
resource of resources.
Mhm. I really like that it's got things for a whole bunch of different levels. It's got if you're some people are text people and some people are video people, it's kind of got something for everybody here. It's really great.
Yeah.
There's a lot of resources for how to get started doing cell painting.
There is, and you know, uh, if it's not clear to watchers of the podcast by now, like we love helping people. We love like these are entirely things that we have been involved in, either, our lab or close collaborator lab. We love teaching this stuff. We love helping other biologists um, learn biology and make discoveries. And so, yeah, it's nice to have it sort of. It was really fun putting this together, curating it in a way like we can take a step back and be like, oh, yeah, like we've done a lot of really helpful stuff. Should make it easier for people to find this really helpful stuff.
Yeah. Well, thank you for making it easy for people to get help.
My pleasure. Thank you for driving the creation of so many of these amazing resources.
Well, so lovely to see you. Talk
You too,
soon.
dear. Sounds good. Bye.